and analysis. So this is where we're going to go with the talk today. I'm going to show you that when we do computational analysis of RNA-Seq, we can still get variations in results even when we use the exact same code and we have a very detailed and accurate standard operating procedure. Then I'm going to show how we can use software containers, Docker software containers, to remove this variability and improve the reproducibility of our results. And as a secondary benefit, of the reproducibility of the software environment, we can do things like more focused optimization. So one of the things that I've done is I've rewritten some of the Python scripts as C++ binaries. And this does actually improve the speed by about twofold at the end of the day. But more importantly, the idea is that if we can get workflows be reproducible and easily disseminated, people might do things like stress test our existing protocols. You might actually try to reproduce things if it's easily reproducible. And so one of the things that came out when we were playing around with different ways to implement the pipelines was we found out that the random unique molecular identifier tags that we were using to normalize our counts were not as random as thought. And that's sort of an interesting result, which I'll go into. And finally, something that we've been working on is a graphical interface to allow people who aren't sysadmins or programmers to play with Docker-based workflows. Because once we have, as you see, once we have these things in Docker containers, they become very easily to assemble into workflows. And the idea is that we want people to be able to play with them, to try different methods, and again, stress test existing techniques and do some reanalysis. So let's start off with what we're looking at. The data set that we're looking at, or the aim is to look for cellular signatures for drug-induced cardiotoxicity. So the basic experiment is a tissue culture experiment where the cells are, let me get my mouse working here, where the cells are grown, differentiated, and then we add different nibs, and they're called nibs because the last name is usually ends in nib. They're protein kinase inhibitors, and as a control, other drugs which are not nibs. Then we take the cells, lyse them, and do RNA-Seq. So basically, as I said, the drug treatments are the control, which is just the DMSO, the actual kinase inhibitor of nibs, and a set of control drugs which are supposed to be non-cardiotoxic, which are frequently taken by patients along with the nibs. So this is the list of the drugs that were examined. So on the left, you see the nibs, as I said, they almost all end up nib except for this monoclonal antibody, which is a mAb, but they're almost all nibs. And then on the other side are the control drugs, which are non-cardiotoxic, at least supposedly non-cardiotoxic. And so what we want to know is what genes, whether it's a differential expression of genes which are in cells which are treated with nibs versus the cells which are treated with non-nibs. So the methodology we're using is RNA-Seq, and I took this figure from 2012 paper, Nature Methods, which takes the basic RNA-Seq method and adds unique molecular identifiers or barcodes in order to aid with normalization. So on the left here, which they had as panel C in the original figure, is a standard RNA-Seq type of protocol. So you take your RNA or cDNAs at that point, take an aliquot, and then before you can sequence them, you have to amplify them, usually with PCR. And then at the end of the day, you take a look at the counts, the number of reads, in each gene or each position. And those, you take those reads to be representative of the amount in your original aliquot. Now, RNA-Seq has a lot of advantages over hybridization methods, hybridization-based methods, mainly in that you can get very, very high sensitivity. You can see very low, low count species of RNA, and also because you have the sequence, you look at variations in both transcripts and very, very minor sequence changes in your RNAs. But in terms of just getting actual counts, there's a big step between the file number of reads, the huge amount of things that happen, and the original aliquot. And you rely on the fact, on all this, these steps in between, not affecting the number of reads that you get. And unfortunately, that's not going to be the case, especially in the application step. There are certain sequences which are going to be amplified more than other sequences. So one way around this is to, before you do the amplification, add a barcode, a unique barcode. So this is a 10 base pair, well, it's a 16 base pair sequence, 6 of them which gives information about what well the experiment is, and the other 10 is a random sequence which is attached. So the idea is that at the end of the day, after you've done all your amplification, you don't count the number of reads, but you count the number of reads and you use UMIS to filter them out. So if they have the same random identifier attached to it, they came from the same molecule. So you can filter out the artifacts from differential amplification. So the other thing that was done that is different from standard RNA-Seq is that instead of taking random fragments which cover an entire transcript, and this would be of interest if we were looking at differences in splicing different isoforms. We instead focus on the pre-prime end. So you get a lot more reads, and if you're only interested in quantifying transcripts, this is a way to increase the depth and decrease the cost of the experiment. So this is the standard data analysis pipeline that we started with. The first step, the input, are FASTQ files from the sequencer. So essentially, a FASTQ file is the sequence it contains a sequence of each read and also a quality measure, so that you know, because there can be sequencing errors. And then we also have these as paired ends. So one file contains the unique molecular identifiers, UMIs, the tags, barcodes, and the other file actually contains the sequence. So the first script goes through and just takes the unique identifier, attaches it to the title line so that we can keep track of the unique the UMI as we go through the processing. And then we do it does an alignment. It calls the BWA binary, which aligns the reads to a human reference genome. So that tells us which gene the read comes from. And then a second Python script takes those SAM files, which have the alignments, and applies the UMI filtering and gives us, at the end of the day, a list of counts. How many normalized counts are in each gene that come from the original experiment. And then this is fed through to a bioconductor package, which actually determines whether which genes are differentially expressed. So most of the computation is actually in this earlier step of the alignment. I found that on our computer it took about a 6-core computer took about 15 hours to do. This only took a few minutes, the second part. But at the end of the day, what we get are a list of differentially expressed genes, which we can give to the clinician and look for and determine gene signatures of which are statistically significant and are associated with the different drug perturbations. So this is from the dTox website. So the level 2 data is the counts data. Level 3's data is a list of differentially expressed genes. So when we started this project, the challenge, the questions that we wanted to answer were basically these three. First, the Sobie group has a very, very good and detailed set of standard operating procedures, which I'll go through next. But the question is, are those, even though as detailed and as accurate as they are those sufficient in combination with the code? Is that enough for a bioinformatician to actually reproduce the analysis? Secondly, the UMI DGA RNA-Seq protocol is fairly new. It's not standard. Will this cause some problems with basically standard processing pipelines? And third, which is sort of related to number one, when people try to run these protocols on their own hardware and with their own operating systems, will this influence the result in any way? So if we take a look at the standard operating procedures, this is the one for how to compare molecular expression. It's actually very, very detailed, extremely detailed. It describes the program that's used and the parameters. It also gives you the exact versions and of both R and the packages that are used. Now, is this enough? So we tried this. Actually, we had a student, Trevor Meiss, who took a counts file and then fed it through the Python or the script that actually determined the differentially expressed genes, and we ran them, or he ran them, on different platforms, and they actually all run. Actually, the ones I'm showing are all actually using different Mac laptops, and we find that if we take a look at it, we do get some small differences in the p-values, and this comes from the fact that even though they're all different versions of Macs, the Mac OS, the earlier versions actually used a different compiler. They used the same compiler that Linux used, a GCC compiler. In later versions of the Mac operating system, however, they changed that. So different compilers will have slightly different implementations of floating point. And it's not that this is a bug, it's just that the standard allows for small variations in representation calculating floating points. And they both adhere to the standards, but they're both slightly different. So that explains why we see some slight differences from the original data, which was, as far as I know, run on a Mac, and this other, our data, which was also our results, which were also run on Macs, but a slightly different operating system, a newer one. So also concerning is that when we take a look at the sorting, we get some differences in the way things are sorted. Now, this is even though that we're using the same operating system. And this actually arises from different localization. So you can, it's not, in the old days we just had ASCII and everyone just used ASCII. Now we have Unicode, and that's localized depending on whether you want American English, Australian English, Canadian English, or Chinese, and that affects affects the way that things are sorted. So this is these are minor changes, but these things can affect the way these minor changes can actually become very, very big changes. In this case, we were lucky, and the changes are pretty insignificant. However, I want to show you a case where these changes in ordering can have a very large impact. And this is actually from an earlier stage in the pipeline, where we're doing the alignment stage with BWA. So at this stage, what BWA does is it takes a read and it tries to map it to a position on the genome. Now, often, usually there's more than one possibility, and then it takes a look at the edit distance and the confidence in each of the sequence in each base pair of the sequence to figure out the best alignment. So it goes and gives you a primary best alignment. And for the most part, that's what we use. We just ignore the other possibilities. Now, however, you can get ties. So the way that you could have the same score, but the way that BWA resolves these ties is to use randomly. And it does, and that may sound that's like a silly way of doing it, but otherwise you would have systemic or systematic type of errors. If you do it randomly, you don't have systematic errors. And to make it consistent, BWA uses the same random number seed at the beginning of each, when it runs on each file. So if you're running the same file and all your reads are in the same order, you will always get the same result. Now, but if your files are slightly different size, or your reads are in a slightly different order, even if you have the same reads, you can get slightly different results. And so let me show you how this very, very small change in resolving tie scores can give a difference in the final ranking of differentially expressed genes. So this was run, so I basically ran the experiment twice, one under the original conditions. And in the original conditions, the files were split into files which are no more than 500,000 lines long. I changed that to 400,000 lines and reran it. So that's all that's been done, and that changes the random numbers that are used to resolve ties. And so when this happens, we actually get differences. Now the gold are when we get differences in ranking. The red is when we get new genes, differentially expressed genes that didn't appear before. So this very, very small change percolates up and does actually change the final results. Now, in our case, this is not something that is dependent on machines, on the machine or the software or the operating system, because the scoring that is used is integer-based, and integers are represent the same on different machines and different compilers. But had that been one could imagine that someone would have a model where instead of using a random number generator to distinguish between ties, that they use some p-value, some double precision p-value, in order to eliminate the random number variables. In that case, we would get something like this. So I mean, this is I just wanted to show that a very, very small difference that could affect ordering can give rise to some differences in your final results. That we can't always assume that our methodologies are robust to these type of errors. That's really the only point that I wanted to make about that. So what are the challenges in reproducing analytical results? Now, we've talked about, one of them is software dependencies and versioning. Now, we can take care of most of that with the standard operating procedures. We've shown how multiple OSs, even different versions of the same operating system, can give rise to slightly different results. And certainly, even when you're using the same operating system, the configuration can, for example, choosing the localization settings can also affect the way things are sorted and potentially affect the results. A further complicating factor is that most bioinformatics analysis nowadays involve not just one tool, but a set of complex tools, each with their own dependencies. So that multiplies the possibility for variations. And finally, sometimes in bioinformatics you actually have tools that use graphical interfaces, and those are very difficult to reproduce because different machines have different ways of representing presenting graphics, of booting graphics to the screen. So our solution is to distribute the entire computing environment and not just the code. Now, we could do this by moving the entire machine, and this is not a completely silly way of doing it. This is actually done when you have a huge amount of data to move that you actually would distribute the disks around. And we also have to remember that originally, when back in the old mainframe days, back in the 60s and 70s, the computing environment and the software were much more tightly coupled. IBM basically used software in order to sell their bundled software solutions in order to sell their hardware. A decision which sort of bit them, which really cost them later on when they decided to license out their operating system on the PC. But be that as it may, this is really not a great way of just moving a small piece of software around to move the entire computer, though it would actually work. It would be 100% reproducible. So in the 21st century, what we do nowadays is we have virtual machines. So if you log on to Amazon and you can get a free instance, free Linux instance, that actually isn't a real computer. It's actually a virtual software holodeck recreation of a computer that sits on top of hardware, whatever hardware that Amazon has available at the time. But the advantage of that is once it's abstracted, once it's in the software, it's completely in software. That is a file that can be transferred, and it actually reproduces the exact environment that the software runs in. So this is actually a viable solution. We can transmit these virtual machines, and they would be an exact representation of the software and its software environment. And this is something that we originally had intended as our way of distributing and reproducibly distributing software. But virtual machines are very, very big, relatively big. It still takes quite a bit of time to migrate a virtual machine and to spin it up. If you were to try this on Amazon or Azure, it'll take you a few minutes to spin up a new virtual machine. And a large part of that is stuff that really doesn't, we don't really have to, which we really don't have to provide because they don't change from virtual machine to virtual machine. So a lot of wastage in this step. So the better solution is to use the equivalent of shipping containers for software. And this is actually a very, very good analogy. The idea of a standard shipping container has really changed the way the shipping industry works. You take the container, you put it on a barge, they're all the same size, so you can just plop them right onto the trucks and they go where they have to go. You don't have to move the stuff inside from one container to another container. They just go, and that makes things so much easier. And so we have the analogous idea for software. Instead of just trying to recompile the code each time we go from a different platform, we ship the entire environment, all just necessities inside a standard container. Once you have a standard container, you have all the tools that can evolve around that container to make it so that you don't have to worry about how to deploy it. You just have to worry about getting your software to work inside that container, and then it can be deployed everywhere. So the standard environment for this that has evolved in the past 4 years is Docker, and it's pretty much everywhere. It's become a pretty standard piece of software on all platforms and most cloud platforms as well. So basically what Docker Container does is it wraps up a piece of software in a complete file system that contains everything it needs to run. The code, the runtime tools, system libraries, everything that you would actually install on a server. It's there already in that container, packaged up, ready to go. And so wherever you run it, as long as you have Docker handling it, it runs the same. It runs the exact same software environment. And we can there's actually a free repository where you can store these images, which Docker provides on DockerHub. So let's just take a look at the differences between a full virtual machine and a Docker container. Basically, on the left we see what would happen if we wanted to have two apps. We'd have to ship two different virtual machines. They'd have to have a complete operating system in each of these, and plus all the libraries and dependencies that they need. With Docker, we can share most of the operating system. The kernel is shared among the different containers, so that makes these containers much lighter. They're much smaller. So, for example, some of our Docker containers are just a few megabytes, as opposed to hundreds of megabytes for a full virtual machine. So, the experiment that we're doing was to take the original pipeline and put them inside each of the steps inside a separate container, and see whether we could increase the reproducibility when we did that. So when Trevor did this, we just focused on this step here, which is where we start with the counts, because for one, it didn't take 16 hours to do. It could be done in a few minutes. And it was something where it was closer to the results that we really cared about, which is the differential expression. So let's take a look at what happens when we use Docker. We put that procedure inside the container rather than running it on the software on each of the individual environments. We run it inside the container. So Docker, here's Docker on Macs, Docker on Windows, Docker on Ubuntu, Docker on the cloud, and Docker on a different Mac OS. And we get the exact same results, unlike the small differences that we saw before. So this actually does work. Now, one other thing that comes out of this, once we get the reproducibility, something that arises from that is that once we've containerized everything, now you can actually do more optimization. You can specify exactly what libraries you want, and you know that will be delivered. You don't have to worry about the user having to have a specific library, or Bioconductor having a specific library available to all its packages before you can do optimization. Now, in terms of C++ here, I was able to replace I was able to replace the Python code with C++. And normally, one of the reasons that you don't do this is because the file manipulations that are required would be very difficult to be made portable. But that's not the case when you containerize it. You rely, as long as it works in your container environment, you rely on Docker to make sure that it will work on the user's hardware or software environment. It completely isolates you from that. So at the end of the day, we started off with shell scripts pipelines. That's how the original pipelines were arranged. They're bash scripts, and modified these in order to use Docker Docker commands. And this is for the first part of the alignment part of the pipeline. Trevor had worked on the analysis part. So this is actually much more computer intensive. So let's take a look at what's actually involved. So this is essentially the original shell script. In yellow are all the parameters, like how many threads you want, how many wells there are, where all the files are. So that doesn't really change much. In green was the original local script. That ran the different binaries that were part of the pipeline. In red are all the things that have to be added for Docker, which is not a lot. So at the top here are basically what the containers are. These are the commands to start the containers up. And then we also have a prefix here so that Docker has to mount inside has to mount the external files so they can operate on them inside the container. And so that has to be added to the files. But as you see, it's not a lot of code. If you organize this properly, it's actually very easy to convert a local shell script into something that uses Docker containers. Now, these Docker containers, they live on DockerHub. We have a repository, BioDepot, and so basically they're images that you download which have the precompiled software and its environment. The code lives on GitHub, and GitHub has in the past 3 years become the de facto standard for distributing code. It wasn't the case in 2013, but it is the case in 2016. Everything is on GitHub now. And so, in addition, in order to specify, in order to create and specify a Docker container, all you need is actually a simple text file, a Docker file. And Docker will, then you can, will take that Docker file and build the container where you can put your compiled code in. And I just want to show you what's involved in that so that's not mysterious or anything like that. So this is a Docker file that specifies the container for the RNA-Seq C++ binaries. Basically, you have a starting image. In this case, this is an Alpine Linux, which is a very small Linux. Now you don't. Now, as I said before, containers actually share most of the operating system, the core, the kernel, but there are always distribution-specific parts of it which it doesn't share, and those have to be provided. So I could have a Ubuntu, start with an Ubuntu set of libraries. In this case, I use Alpine because it's much smaller, it's only like 5 megabytes. And then I run and install inside the container all the necessary libraries I need for my my C++ code, including the file system. Now, if I were to try to do this without a container, I would have to have the person install the boost libraries externally. But Docker handles all that, the file manipulations. I don't have to worry about that. And the rest of it is just some customization which I can add to the container. In this case, I have 2 binaries, one for 96 wells, one for 386 wells. So you can do some customization on top of the actual binaries as well inside the container. Now, I wanted to bring up to show that this is something which is actually very useful. The use of containers, especially for deployment in the cloud. I talked to some HPC people at a Docker conference, and they were saying that it can sometimes be very difficult to provision a large HPC cluster, especially with normal pipelines or scripts that use Python, and it can take a lot longer than it actually takes to run the job on the supercomputer. But when you do it with Docker, it becomes much easier because, as I said, Docker only takes a few seconds to spin up if you have a small container. And so this group, David Hoster's group at Santa Cruz, took this to the extreme and reprocessed 20,000 RNA-Seq samples from TCGA. It took 4 days for them to do, and it only took $1 per sample. And this was all done using Dockerized pipelines so that they could rapidly deploy these on the same pipeline in parallel on many, many, many different cloud instances. So this is a very, very useful thing, especially for parallel HPC type of applications in the cloud. Now, I want to get back to something a little bit more for local that I found when doing the re-implementation of the pipeline C++. One of the things I was able to do was re-implement the original algorithm where basically if two reads map to the same position, then they have the same UMI, then you filter them out. That was the original implementation. The Python script, because of the limitations of Python, in order to save memory, made the approximation that if they map to the same gene and the same UMI, then they're the same fragment. Now, that's true if they map to the same position. And so this shouldn't make a difference if there are enough different UMIs, if they really are. And there's, with 10 base pairs, we have about 1 million of them, so you wouldn't think this makes a difference. But actually, it does. So if I just take a look at the entire experiment, I can take a look at the first, this is the entire experiment, and I just take a look at the first 600,000 reads, a fairly small number of reads. If I don't do any UMI filtering, I can map 366,000 of them to the reference. And then if I do filtering with UMIs which are at the same position, it's almost the same whether I do based on whether the UMIs map to the same position or to the same gene. However, if I do all the reads, now this is the exact same experiment, but I'm doing all the reads now, I see that when I do the gene specific, I get a lot fewer counts. And this is because I'm getting the same UMIs which are probably which are mapping to different positions within the gene. And this shouldn't happen when you have 1 million of them. Now, of course, when you take a look at how this affects the top 40, the list of differentially expressed genes, makes a huge difference. And you would expect that. I mean, you take a look at the number of reads mapped. You have more than twice the number of reads mapped when you're doing position filtering as when you're doing gene level filtering. So why is this the case? So if we take a look, the entire experiment is roughly 5 million reads per well. It works out to roughly 500 reads per gene, and some genes are going to have like 100 times this. So there could be more. But still, we have 1 million possible UMIs. So this still we shouldn't get to two of them mapping to the same, or randomly mapping to the same gene. But that assumes that they're randomly distributed, and they're not. And this is where when you take a look at the distribution of the UMIs, you find that there's a heavy, at least in this batch, there's a heavy bias towards T's. We expect 300, roughly 300 if it was uniform, but the most common one is 63,000. And there's a bias against C's. But so what's happening is that we're not getting actually an effective 1 million random tags. We're getting a much smaller population of bias, of heavily biased UMIs, which is causing this unexpected problem. So so what came out of this is that we found out that we're getting more of these duplicates mapping the same gene than we would expect because of a non-uniform distribution of UMIs, and also because the reads aren't exactly 500. There are going to be some which are highly expressed genes which are going to have 100 times that. And those cases are going to have a lot of duplicates which artificially depress the counts. And because the likelihood of having these duplicate UMIs mapping to the same gene is going to depend on the number of reads, this is not normalized out when you do the differential expression calculations. The solution, once you know the problem, it's not difficult. It's actually quite easy to exclude prune these these overrepresented UMIs, and use position level filtering so that it's much less likely that you get two duplicate UMIs from different fragments. So finally, I want to move on to this a final thing, which is something our group has been working on, which is to adapt graphical applications to be used with Docker. For the most part, this has been something which has been neglected. Docker has been used mainly for command line applications. So one of the things that we looked into was to provide a minimum set of software dependencies so we could export graphics. And it turns out this is a paper that came out in April in PLOS One. And so all that we have to do is we have to add an X11 server inside the container. Linux for them, what still does, uses X11 libraries to commands in order to render graphics. And then you have to have an X11 client on the server side in order to render the graphics locally. So these commands are passed through through an SSH tunnel, and then they're rendered locally. But this is a fairly decent solution, but it requires the user still to install their own software, which is something we didn't like. So we went to another solution, which is to use VNC, a VNC-based solution. In a VNC-based solution, the difference is that we don't pass the actual commands. We render the actual screen in the container, and then we pass the screen out. Now this was, the reason we didn't do this originally was because back in 2010, in the old days, this was not a very good way of doing things because you passed a lot of information across. But things have really gotten better. And the other thing is we don't have to have, the main advantage of this is that we don't need a client. We basically have a mini web server which we can deploy in the cloud, or we can deploy locally, and then we just use a browser which uses a HTML5-based NoVNC client in order to hook up to the container. Then we can also use the fact that it's a mini web server to do validation. So this can be done securely by using a third party to provide validation. And this is something which should be coming out soon in GigaScience. We just resubmitted this. So one of the things that we want to use this for, and I'll demo in the last little bit, is we want to have a nice graphical interface so that people can use the fact that these Docker containers are modularized and construct their own pipelines and run their own pipelines without actually having to write a shell script or a Python script or do any real programming. And so we found a very nice interface, an open source interface, OrangeML, which is for machine learning. And we've wrapped it inside a container which supports graphics. And I'm going to demo that right now and show you how that works. Now let's go and, all right, so this is being run on Windows right now. Let me quit this, and this is always the thing with live demos. Maybe we can just go to the and reload. All right. Okay, there it is. So this is actually a running the OrangeML app in a container on Windows 10, and it's a really nice app. Let me just start it up again. I'll start from the beginning. Discard all this, and our interface provides a very minimal virtual machine, essentially, which wraps the application. It'll run exactly the same. We've tested this on Windows, Linux, Mac OS, and also on the cloud. So this is a very, very nice interface for machine learning, and we've adapted it for our own Docker pipeline. So this is the detox, the components of the original detox pipeline. So we have the alignment step, and we have the analysis step. So we can put these out and make our little pipeline. So the alignment step feeds into the analysis step, feeds counts in. We have to feed a directory in there. We can have the same directory feed for the references and sequences, or we can have a separate one. So let's do a separate one, and you'll see what that's pretty cool. And we put another directory here. So that directory is for the human reference. This, this. Come on. And this is for the actual sequences. It knows that that's the only possible one. And then we can also hook up with the tools are locally available from RNaseq and go out to a feed the results to a data table and then save that data to a file. So so we can actually run this if we wanted to and save this and send to somebody else. So this is mostly the work of Trevor Meiss, who has implemented the new widgets. We're getting so the idea is that we're going to have a whole bunch of widgets which you can just plop in. So instead of using the detox alignment, you can put in Starliner. Instead of using or you can use some of the C++ based containers. The idea is to take this customization and put it in the hands of people who are normally programmers and hopefully get more people to use this type of thing. So let me just finish up the talk here. And go to slideshow from current slide. Okay. So the takeaways are, we've shown that the RNA-seq analysis can give variations, even the same code and operating system. We've shown that the Docker software containers can remove this variability and allows for easy deployment, especially on the cloud, which can be very, very useful, especially for HPC type of applications. I think one of the things which I hope to get across is that I think potentially the ease of mix matching components or workflows because of Docker, I think this will be transformative. I think that this will allow people to reproduce and quickly analyze and workflows and reanalyze data. And things like this UMIS tag, these little artifacts which pop up, will pop up when we do that type of, we allow people to do that type of digging. And finally, these are the people that did a lot of the work. Eric, Sobie, and Granville were very appreciative that they allowed us to take their operating procedures and software and basically kick the tires and look for defects. Daniel Cristianto was one who basically started this, our Docker project, because he wanted to work with VMs. It was with Ka Yee's husband who said that VMs are so 2010, we should be using Docker, which is all the cool kids use. And we did. Sung Lee started the GUIdock stuff basically because I told her it's very easy to add GUIs, and it turns out not to be, and so we wrote a paper on it. Trevor did most of the stuff with the original reproducibility. We show those minor differences, and also the OrangeML Python stuff is mostly due to him. He wanted to do this originally in Java with a much simpler interface, but I think he changed his mind once I mentioned that there's a $45 million startup doing a very similar interface. Varun provided the original no VNC code, which we use to wrap our Orange ML interface. He educated me on the advantages of VNC, and not only did he educate me, he came up with a implementation within a week after we talked about it. So the VNC implementation is mostly due to Varun, and of course I'd like to thank Ka Yee Yeung, and who is the head of our group. And if you've seen a pattern here, it's that people seem to go on different paths from what they started with, and Ka Yee's very, very supportive of that. So we get to play with a lot of cool toys while we're doing our work, cool computational toys, and it's really helpful to have someone very supportive of that. And if you want to see some of our cool toys, you can go to our GitHub, where we have a lot of our software and Docker images. And I think that's it for time.
Thank you, Ling, for presentation. Do we have any questions from audience?
Hi, this is Yuguang from Mount Sinai, Guangzhou. About that non-uniform distribution of the UMI tags, I'm thinking, this non-uniform distribution can be caused by the PCR process, or
that's a good question. I don't know. We'd have to take a look at more samples where this is consistent across different. I mean, it could be something in the PCR process that the poly-Ts are more easily amplified, but it could also be a very small difference when they synthesize these UMIs. I really don't know.
Okay. Okay, got it. Yeah. All right. Oh, so all the code on the BioDepot in GitHub, right? So I can get from there and deploy it on my machine, on my computer using Docker. Right? Because I just downloaded Docker. Yeah, I want to try it out.
Yeah, the script should run right away on locally. I ran them in the cloud because it actually takes quite a while to do the alignment step.
Yeah, of course. Yeah.
Yeah, and yeah, but they should run on your laptop as well if you have enough disk space. It takes a lot of disk.
Oh yes. Okay. The last question is you wrote a C++ program to replace the Python script, right? And what? So I'm looking at the GitHub homepage. It seems like your C++ program can use much less memory than the Python programs.
That's right. And that's basically not so much the Python per se, but the main source of the memory usage in the Python code was using a text-based hash rather than a perfect hash. And so when you change it to a perfect hash, you save a lot of memory, and then that's why you can do the position level filtering. And it was perfectly reasonable to do the text hash, to be honest, because if the UMIs had been more uniform in the distribution, it shouldn't have made a difference.
Okay. All right. Yeah. I will try a program, see how it works.
Great.
Yeah.
Well, if there are no further questions, thanks again, Ling, for presentation, and everybody else for attending Data Science webinar. We'll reconvene next time.